Hello, my name is Dr. Peter Kopko. I am a chiropractor and practicing dark field microscopist. What I'd like to discuss with you today is the effects of dehydration and acidity in the body on some dark field parameters. I'd like to take a look at this chart. Normal blood are nice round red blood cells as seen on dark field microscopy, 6 to 8 microns in diameter, and they would be free floating in this plasma. When red blood cells start to lose their net negative polarity, they start to stick together. This process actually starts in a phenomenon that's referred to as protein linkage, where the red blood cells start to hook together much like linkages of sausage. These then spin on top of each other and stack up. The next phase is called rouleau. It's French for a stack of coins. If it further coagulates, it becomes a ball of cells, which is referred to as erythrocyte aggregation or blood sludge. There should be a net negative charge on the surface of a red blood cell. When the body starts to become overly acidic, this net charge is changed to a positive. Negatives attract positives and the cells start to stick together. By drinking alkaline, ionized, microclustered water, the ionization process to the water creates a net negative charge to the water. You're reintroducing electrons into the system, which helps reverse that polarity switch on the surface of a red blood cell. We have in experiments here at Life Ionizers taken people, looked at their blood, saw that it was in a coagulated state, had them drink 2 of these containers, which is approximately 1.5 liters of water, and 30 minutes later looked at their blood once again under the dark field microscope and saw how the cells had normalized. And we're going to demonstrate that for you today. We'll place it on the slide. Using this chart will help you view what you're looking at here. Your cells are all stuck together, much like depicted in this slide right here. You have erythrocyte aggregation. What we're going to have you do is to drink 2 of these, which is approximately 1.5 liters of water, over the next 30 minutes. We're going to set a timer, have you come back and recheck.
Okay.
Here we go. All right, thank you. Appreciate it. As I stated previously, the process of red blood cells sticking together actually starts with a process called protein linkage, where the ends of each of the red blood cells stick together like links of sausage. If the problem continues with the reversal of the polarity on the red blood cell and more acidity in the system, the cells start to stack up, and that's referred to as rouleau. And that's what we can see has occurred in Mr. Kabatis's blood. We're going to take another blood sample, and once again, his original blood was stacked up and bunched up, and we're now hoping to see that it is completely disassociated, free-flowing, nice round red blood cells. We'll place it on the slide. So we can see that his cells have completely disassociated after drinking the water, which once again has restored the net negative polarity to the cell of each and every red blood cell. In 2003, the Nobel Prize in Chemistry was awarded to a Dr. Peter Agri. What he discovered was the way that water molecules move into each and every cell in the human body. He termed it an aquaporin. It's a pore on the surface of the cell where water molecules move through. A water molecule is the smallest molecule in the human body, and these pores are exactly the diameter of one water molecule. I have an animation that I can show you. This is a cross-sectional view through a cell, and the actual aquaporin, the pore, is right here. And this is going to show how a water molecule approaching this pore will eventually move down this channel and into the cell. Each one of these molecules is an H2O molecule. The oxygen is red and each hydrogen is gray. I'm going to start the animation. If you follow this yellow marker right here, you can see that things are eventually moving down. Each cell approaches here and then travels down the pore. Now, our alkaline ionized microclustered water, we believe it accelerates this process in two ways. The clusters of molecules are less. Normally in tap water, there are 11 to 13 to 15 water molecules in a cluster. When water becomes alkaline and ionized, it's reduced to 5 or 6. So there's literally a quicker availability of each water molecule to start moving down. Secondfold, there are proteins along the lining of this channel that have a specific net positive charge to them. And when a negatively charged water molecule approaches it, which is created by alkaline ionized water, we also feel that it will accelerate this passage. And once again, here's this yellow marker, and as you can see, everything is moving essentially south and into the cell.